Okay. Very good evening, my dear friends. Today's our eminent resource person, Dr.A. Madhuri Madam Garu, Assistant Professor, Department of Microbiology, one of the important, one of the prestigious institution in Hyderabad, that is GDC for Women, BEGUMPET, Autonomous College, Hyderabad. Madam has huge experience in the field of microbiology and also plant sciences. So Madam is going to explain today's one of the important topic, that is transcription. Madam, please start your session. Please welcome.
Yes. Good evening, students. Here, whatever PPT I'm going to show, in that structure of RNA polymerase is not shown. That is why I'm showing it specially. Here, if you focus, I'm not able to maximize this structure. Just you see the subunits in the RNA polymerase, alpha 2, beta, beta dash, omega. This is RNA polymerase. Okay. One more subunit will come and join this. That is sigma subunit. Are you able to follow? Am I audible to all of you? Yes, you have to respond. If you respond, I can
respond. Shravanti, Rakesh.
Whoever are there, please respond, ma. Okay. So, what are the subunits here? Alpha 2, beta, beta dash, omega. This is core enzyme. This is called as core enzyme. If sigma join this, if sigma join this core enzyme, it will become holo enzyme. Okay. It will be asked in the entrance, right? So we will start with the transcription now. Okay. Okay. Sorry for the disturbance. So this is the structure of RNA polymerase. Alpha 2 alpha subunits, one beta subunit, one beta dash, and one omega, and one sigma factor. This is RNA polymerase of E. coli. Okay. So without sigma factor, the enzyme will be called as core enzyme, and with sigma factor, it will be called as holo enzyme. Important bit for your entrance. Okay. So what is the role of sigma? It is used to trace the promoter sequence. Just remember, it is used to trace the promoter sequence. Okay. Is this clear? RNA polymerase? Yes. Okay. So we'll start with the session of transcription. In this transcription, you have to understand what is upstream and what is downstream. Okay. In this picture, if you see, uh, there are three partitions. Okay. One is promoter, second one is RNA coding sequence, and the last one is terminator. Right. The sequence present towards left side or 5' side is called as upstream sequence. The sequence present towards 3' side or terminator side is called as downstream sequence. Hope I am clear here. What is upstream sequence? What is downstream sequence? Okay, upstream sequence is present towards your left hand, and downstream sequence is present towards your right hand. Clear, ma? Next, gene structure. As we have seen, there will be promoter sequence, RNA coding sequence, and terminator sequence. Now, what is promoter sequence? Promoter is the region where RNA polymerase will come and bind. Okay, what is promoter sequence, ma? It is the sequence where RNA polymerase will come and bind. Before this, you have to understand one more concept, right? Replication. What is the enzyme required for DNA replication? Anybody? Answer fast. What is to be synthesized in replication? Sir, am I audible?
Yes, madam, you are audible.
Okay. Uh, Nobody is interacting actually. No problem. So, in replication, DNA has to be synthesized. So, the enzyme name is DNA polymerase. Okay, in transcription, what is transcription? It is the mechanism of RNA synthesis from the sequence present on DNA, right? From DNA we are getting RNA. So this is transcription. So what is to be synthesized in transcription? It is RNA. So the enzyme name is RNA polymerase. You need not get confused in the names of these enzymes. DNA polymerase is for replication and RNA polymerase is for transcription. And what is promoter? Promoter sequence is the sequence where the RNA polymerase will come and bind. Okay, this much you have to remember. Next, RNA coding sequence. This is the actual sequence uh, uh, should be present in the mRNA. And terminator sequence or terminator region is the last region where the gene sequence tells the transcription machinery that it should stop. Okay, so the transcription process will stop at the termination sequence, right? And as replication is having three phases, here transcription is also having three phases or three steps: initiation, elongation, and termination. And here, just now we are talking about promoter region. Promoter means that it is the region where the RNA polymerase comes and bind. Here you see, this is the RNA polymerase, the bigger segment, and here they are showing the sigma segment separately. Okay, so when the sigma comes and bind to RNA polymerase, then only it can trace or it can track the where is the promoter region. What is promoter region? It is the region where RNA polymerase should bind. Okay, and this is the sequence. Here they are showing -35 sequence and -10 sequence. Okay, this entire sequence is promoter sequence. Now what is promoter? Where it is present? Yes, it is present on DNA. Yes or no? So this sequence is present in this. This is the promoter sequence, and on this promoter, this RNA polymerase is coming and binding. Now we are studying the structure of promoter sequence. You have to remember, in the promoter sequence there will be two consensus sequences. Okay, what is consensus sequence? Is it is a sequence which is common in majority of the organisms. Okay, so in these two are consensus sequences. What are they? -35 sequence and -10 sequence. This Pribno box is called as -10 sequence. Okay, so here this is the sequence of -35, that is TTGACA, and -10 sequence is TATAAT. Right? Hope I am clear here. And this is the mRNA start sequence. mRNA start sequence. Okay, um, means the synthesis or the first base will start from here. So when you going back, when you going to the left side, the bases are given minus sign, and when you go beyond this +1 side, the bases will be given plus sign. Okay, so this is nothing but upstream and downstream. Next, coming to the initiation, as I told you, sigma factor should bind to promoter sequence. It should bind to polymerase, and then the entire polymerase will come and bind to the promoter sequence. Okay, Next, so same thing here, core enzyme binds to sigma factor and promoter, but DNA is still closed. Now you have to understand when sigma binds to polymerase, the core enzyme is converted into holo enzyme. Now in this initiation step, there are some stages. Okay, here you have to remember closed promoter complex and open promoter complex. Open promoter complex, closed promoter complex and open promoter complex. Okay, so in closed promoter complex, what is happening is RNA polymerase has come and bound to DNA promoter region. Okay, but the DNA here is not unwinded. It has not opened. Okay, so that is why it is called as closed promoter complex. The DNA is not yet opened, or the hydrogen bonds between the two strands of DNA are not broken. That is why it is called as closed promoter complex. And this part here you see DNA has unwounded. Okay, DNA has opened, right? That is why it is called as open promoter complex. These two terms are important, right? So in initiation, we have seen closed promoter complex and open promoter complex. Okay, now after opening, what happens? Core enzyme, once it uh, as the initiation continues, RNA polymerase binds more tightly to the promoter, accompanied by local untwisting. At this point, RNA polymerase is correctly oriented to begin the transcription at +1 site. So when RNA polymerase sits at correct position, then the transcription starts at +1 site. Okay. After the RNA polymerase finds the promoter region, the sigma factor dissociates from it, right? Only core enzyme will remain. Now, core enzyme moves along the along the DNA to form RNA segment. Okay, when transcribing, RNA polymerase interact with DNA sequence, forming transcription bubble. As you have seen in replication, there will be a replication bubble. Here there will be transcription bubble. Okay, see here, this is the DNA, blue color, and red one is the RNA which is being synthesized. Okay, you very well know what are nucleotides present in DNA, what are nucleotides present in RNA. Only there will be the difference between thymine and uracil. Rest all will be the same. So as the cell gives these NTPs, nucleotide triphosphates, the synthesis of RNA will continue, and this is RNA DNA hybrid present here. The synthesis is going on, and the round bubble is called as transcription bubble. And what is the direction in which transcription takes place, ma? It is always, this is important bit, it is always 5 prime to 3 prime. The direction of replication is 5 prime to 3 prime. Direction of transcription is 5 prime to 3 prime. Direction of translation is also 5 prime to 3 prime. Never confuse with this. Okay, always the direction is 5 prime to 3 prime. Now, here RNA polymerase is also having proofreading capability. Means it can check if there is any wrong base incorporated, it can go back and remove that base and add new or correct base at that site. So this was about initiation, elongation, and last part of transcription is termination. Okay, here in termination means the gene sequence which has to be synthesized into, or which has to be copied into RNA form is done. Now the chain or mRNA has to dissociate from the DNA. So that is called as termination. Okay, so termination can be of two types. One is row independent termination, and the second one is row dependent termination. Row dependent and row independent. Okay, now we are talking about row independent termination. Here, what happens is at the termination site, there will be palindromes. What do you mean by palindromes? These are the oppositely complementary sequences present at a distance, 1 cm. Okay, 1 cm. 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 1 in opposite directions. Here, the basic mechanism or the basic purpose of this presence of palindromes is to form this stem loop structure. Okay, when this stem can be formed, when this bases can be attached, when there is complementary sequence. If you see GC, there will be a bonding. GC, there will be a bonding. AU, there will be a bonding. CG means these two are complementary, means these two sequences are complementary. Okay, so these complementary sequences or palindromes, they are present in the termination sequence. So as the RNA is being synthesized, immediately RNA will form this stem and loop structure. Okay, so due to this formation of stem and loop, this will drag or this will pull out from the DNA, and the RNA synthesis will stop there. This is row independent termination. Here row factor is not involved. Row factor is not involved. That is why it is called as row independent termination. Here only the sequences are present in opposite direction, or the complementary sequences are present in opposite directions, or they are also called as palindrome. So they, after the synthesis of mRNA, it will form stem and loop structure. So with the formation of stem and loop structure, it will come out of the as there are more uracils, it will, the bonding between DNA and RNA will be weak, and then RNA molecule will come out of this process. Next is row dependent termination. Okay, so here what is row dependent termination is, row is a protein. Row is a protein. What it will do is, here actually normally terminator sequence is rich in cytosine and poor in guanine. So terminator does not form a hairpin loop here. Here in row dependent termination, there will not be formation of any hairpin loop structure. Here a row protein is involved. It will come and bind to the RNA. So what is the function of row protein is, it is having some helicase kind of function. Okay, in replication you might have studied helicase, it will unwind. Okay, so here also row protein will break the bonding between DNA and RNA hybrid, and then RNA will be released from the process, RNA polymerase and row. Then after the RNA has come out of the DNA, then row protein will get dissociated. Okay, so this was about transcription in prokaryotes. Hope I am clear. If you want, I can revise quickly. Yes, Rakesh, Shravanti, is that clear, ma?
Clear, ma'am.
Shall I repeat?
Yeah, repeat, madam.
Or shall we stop? Okay. So how many stages will be there in transcription? Initiation, any process. Initiation, elongation, and termination. What is the enzyme required for transcription? Rakesh, what is the enzyme required for transcription? It is RNA polymerase. Okay. And what will be the subunits in RNA polymerase? Alpha 2, beta, beta dash, omega, and sigma. Okay. If you remove sigma, the enzyme will be coenzyme. If you add sigma, the enzyme will be holoenzyme. This is the difference between coenzyme and holoenzyme. Now, what is the function of sigma factor? This helps that coenzyme or the RNA polymerase to track or to trace the promoter to DNA, and then it will move throughout the DNA. Wherever it finds that promoter region, it will stop. Okay, so it has to identify that promoter sequence on the DNA. That identification will be done by the sigma factor. And then once it identifies that promoter sequence, sigma factor will come out of that core enzyme, and then core enzyme will start transcription. Okay, and the sequence present on left side to the +1 site is called as upstream, and the sequence present towards the right side of the +1 site is called as downstream sequence. Okay, and then we have seen in transcription there will be initiation, there will be open um, sorry, closed promoter complex and open promoter complex. Closed means here the DNA is not unwound, and in open promoter complex the DNA is unwounded. So, okay. In transcription elongation, we have seen transcription bubble will be there. New RNA will be synthesized here, and there will be an RNA DNA hybrid, and the direction of transcription will be in 5' to 3' direction. Okay, and it is also having proofreading capability, and then termination is by two methods. One is row independent termination, and the other one is row dependent termination. Row independent termination, there will be hairpin loop structure formed due to palindrome sequences present in the termination region. Okay, so due to this hairpin loop structure, the RNA will be pulled off from this DNA RNA hybrid, and it will come out. And in row dependent termination, there will be a protein called as row protein. The function of this row protein is helicase function. So it will cleave, or it will break the bonding between DNA and RNA, and then it will release the RNA. After the RNA comes out from the DNA, the row factor will dissociate from the RNA molecule. This is about transcription in prokaryotes. Based on this, you can build your subject more. Sir, we can stop the. Hello. Okay, ma, we'll stop this session here.
Okay, thank you very much, madam. Nice presentation in this today's class, one of the important topic in molecular biology. Thank you very much, madam.